This unit, this series, this day in Jan Plan has traditionally been saved for the special senses. I save it for the special senses. And, or not just the special senses, but sensory organs in general. And with time, because, that covers, I could probably spend a whole semester, like a full semester, just talking about all of the sensory receptors and their functions in depth. But with time, I've kind of whittled it away. I don't actually talk about taste and smell nearly as much as I have in the past, or as I'd like to. And I focused on sight and hearing, because those are really two of the most profoundly awe-inspiring, at least to me. So it'll be mostly sight and sound today, and some balance. The next few slides are really all that remains of the larger unit that I used to crash through at a breakneck pace. I had mentioned Meissner's corpuscle, I think yesterday, when I was talking about the different types of neurons. And I'm just putting these out because my TAs must not have remembered that they were on this unit. But you can see them on those two models that I stuck out there. Meissner corpuscles. Anybody remember what kind of information this is returning? What specifically? Yeah, Liam. Yeah, fine touch, vibration. Yeah, that's right. Soft, fine touch. That's it. And these sort of collagenous bundles that wrap around the terminal nerve fibers are embedded in the dermal papilla that underlie the epidermis. So the dermal papilla are these ridges that you see there, and very easy to identify on a slide. So there's a bunch of slides over here near Amanda and Grace. You can take a look at them. I've put them on slides in the past, not every year, but it's a really easy thing to point out. So yeah, there isn't really much salient anatomy beyond identifying them. Next is the Pacinian Corpuscle. Now, a Pacinian Corpuscle, also on those two models I laid out there. In the model, you'll see they look kind of like a green onion on the lower left-hand side. Ooh, I forgot I had this year. I bought myself a third skin model, 3 benches, 3 models. My goal is to get 3 of everything. I haven't even. So here would be a Pacinian Corpuscle, and this would be a Meissner corpuscle. Whoever wants to on that one. Yeah, the Pacinian one is towards the bottom, sort of that multicolored thing, and then it's red. The red ones in the papillary layer are the Meissner corpuscles. The Pacinian corpuscle in the histology slides are pretty easy to identify. They're at the boundary between the reticular layer of the dermis with the dense, dense irregular connective tissue of the reticular layer of the dermis, and then this is hypodermis with all of its fat. You can see all the fat vacuoles down here in the hypodermis, and the Pacinian corpuscles live right at that boundary, right at the junction between those two layers of the skin. In cross section, they kind of look like an onion. If you were to cut an onion across its latitude, just all these like annular rings, and right here at the center of one, this is 2 of them. At the center of it is the nerve fiber that runs right down the middle. Okay, so this one we're cutting it. It's the nerve fiber's coming out at you. We cut it crosswise. This is kind of a longitudinal section obliquely across the side of one. so you can't actually see the nerve fiber in that one. Here's an exquisite section of one where we can see right along the length of the nerve fiber, and the layers of the Pacinian corpuscle are like that. Now, what was the Pacinian corpuscle? What modality was that returning? What kind of information? You guys remember? Deep. Yeah, deep pressure and vibration, exactly. And so if it's returning vibrational information, could you guess whether it's going to be a tonic or a phasic? Phasic, great. And why do you say that? Yeah. So okay, go on. Sorry.
No, I was
Yeah, speculate, please. Tell me more. I mean, I kind of explained it from one direction. I just explained it from the deep pressure side, why it's phasic, but I didn't talk about it from the vibrational aspect of it. Yeah. So it tends to be, if you think about this, and the rationale will make sense. There will be a logical sense to it, but rapidly adapting receptors are good for returning vibrational information because they reset themselves quickly and are able to register a new stimulus. All right, so that sets them up for vibrations. If it's a tonic receptor, it's not going to really be able to register a vibration because it's been turned on and it stays on. So further stimulation is not going to register in terms of stimulating that nerve fiber. Does that make sense? Okay. So deep pressure, which is something that we want to be phasic, because when you don't want to be constantly aware that you're sitting on your butt. You just sit down. I feel that I've sat, and then you sort of forget about it. Or the pressure in your feet. You're not thinking about that consistently. All right. So that's all that I'm going to talk about in terms of tactile receptors. Olfactory epithelium. So let's see if I have a better slide. This actually is not that good of a slide. Okay, this is a little better. Yeah. Olfactory epithelium is the mucosal membrane that is lining your nasal passages. All right, and there's a couple different types of cells. We're going to have the ciliated, the simple columnar epithelium. All right, ciliated columnar epithelium, adjacent to what are called these goblet cells, and we can see the goblet cells all over the place right here, embedded. So in between are these columnar epithelial cells, a simple layer of columnar epithelium with the cilia all over it. This is a transect of a little fold in your nasal passage. And then these goblet cells are, it's a really evocative name because those goblet cells are indeed kind of like goblets that are filled with mucus. Who doesn't love a goblet full of mucus when they're trying to smell the roses? They release that mucus onto the surface, onto the ciliated surface, and that is what dissolves the odorants that are then able to come in contact with the sensory neurons of the olfactory bulb that enable us to smell. Smell is an incredibly complicated sense that sort of deserves more treatment than I'm giving it, but that's about all I'm going to say because of lack of time. Sadly, smell is not even really treated in medical school very much, so you have to, if you want to learn much about it, you'll have to forge out on your own to find the information. And Bowman's glands. Am I likely to ask Bowman's glands? I'm unlikely to ask Bowman's glands, so let's not press that. Taste buds and papillae. Okay, so with this slide, I have a model which I have not yet incorporated into this slideshow. I meant to do it this morning if I was able to get here earlier than I did, but the snow prevented that. This entire structure here is a papillae. You can see papillae, you can visibly identify papillae. And I should do this. I should do this in the class. I'm going to revise the lab next year to make this a more fun one, more interactive. What you can do sometime, you can do this at home. This will be a fun thing. You can either get some blue food coloring, or you can get yourself like a blow pop or something like that, like a blue blow pop, some kind of a sucker that's blue. The goal here is to dye your tongue blue. All right, and then you take either like a hole punch in a piece of loose leaf paper. You can just tear out the corner of a loose leaf paper that has a hole punched in it for putting it into a spiral notebook, and you stick that on your tongue, and it turns out that these papillae are not stained by the blue dye. They don't absorb blue dye, and you can, if you have somebody with a magnifying glass and a light shining on your tongue, you can stick the circle over the tongue and count the number of pink papillae that show up on your blue tongue. Next time you get your tongue all stained blue, look at it in the mirror maybe. You can do it yourself. You just look in the mirror. If you have 15 or less within that area of the circle, standard hole punch circle, you have a normal taste sense. If you have above that, anywhere up to from 50 up to, or up towards 50, anywhere from 15 to 50 is considered to be what's called a super taster. A super taster. You have a much more acute sense of taste than other people. So people's ability to taste actually widely varies in the population. A lot of people aren't aware of that. And it turns out that women have a much higher ability to taste than men. They have there's a much higher percentage of women that present as quote unquote super tasters. I've heard all kinds of different rationale for that in the past, but it's kind of an interesting self-experiment that you can do sometime. Just dye your tongue blue and see what's up. Super tasters tend to be people that find certain things much more bitter than other people. So, for example, they may find strong coffee, unsweetened coffee, to be something that they find a little too bitter for their palate. Other such things, right? But within a taste bud, within a papilla, you're going to have these taste buds, these little taste buds that you see along here. So here's a cross-section, a bunch of papilla, and the taste buds along the lateral aspect of those papilla. You can see some papilla on this model, as well as a taste bud with all of the different sensory receptors in a single bud. So a bud itself is a bundle of sensory receptors. Within a single bud, you're going to have, usually, you're going to have sensory receptors for all of the taste modalities. And I had mentioned them yesterday. Sweet, sour, bitter, umami, salty. I may be forgetting one, but I think that's it. Yeah.
How come when stuff sinus infection?
Yeah, it's not that you can't taste. You can, in fact, taste. It's that the experience, the sensorial experience of eating involves a lot more smell than you think it does. And so, for example, when I list sweet, sour, umami, salty, and bitter, right? Those are very distinct experiences, but that's not like saying roasted almond, right? Or burnt cheese, or eating a lavender cookie or something like that, right? Those are pretty sophisticated experiences, right? Sensorial experiences. And your sense of smell is profoundly sophisticated. It's profoundly sophisticated. The range, the complexity of odorants that our body is responsive to is pretty dramatic. So when you have lost the ability to engage the sense of smell as you're eating, you've kind of reduced your experience down to the much more monochromatic experience that is being given to you by your taste buds. All right? So you can think of it like this, like your taste buds are giving you the like basic quality of what you're eating. Is it sweet? If it's sweet, then it's some sugar, and maybe you want to eat that, right? Because evolutionarily, we didn't have access to as much sugar as we do now, and you want to put, normally sugar is a great source of energy. Or is it umami, which means we got some protein? It's just glutamate. It's just glutamate, right? But if you got glutamate, then you got protein, and that's something your body may need. Is it salty? Your body needs salt, right? Is it bitter? If it's bitter, then perhaps it's a poison. A lot of things that are bitter are not good for you, which is one of the reasons people hypothesize that women are more super tasters. They have a higher, like women who are super tasters, or people in general that are super tasters, are much more sensitive to bitter compounds. And they think that was women because women historically have been the ones preparing the food and maybe gathering. They were the gatherers of the hunter-gatherer dynamic. So it was the responsibility of the woman to be able to make sure that the food was not poisonous. I don't know. Anyways, but then so the taste buds are telling you the basic quality of the food you are eating, but then your olfactory sense is giving you the palatability of it, the like complexity of the food that is making it more appetitive to you. Does that make sense?
Yeah.
It's really interesting topic. Really interesting, I find. If you want more, you can take the biochem food class I teach in the fall. All right. So on to the eye. First, the external anatomy of the eye. The eyelids are called the palpebrae, or plural palpebrae. And where the two palpebra meet is called the canthus. So there's a lateral and a medial canthus. A lateral and a medial canthus is where the eyelids meet. We have eyelashes along the margin of either superior or inferior palpebra. And the space between the eyes, that you probably have never thought about it like this, but the space between your two eyelids, your two palpebra, is called the palpebral fissure. All right, this is the palpebral fissure. It's the space that our eyes are able to look out of. All right. Next, if you look at anybody who spent any time looking at their eyes closely in the mirror, will have noticed the lacrimal caruncle. It's that sort of fleshy colored flap of skin that's on the medial margins of either eye that just covers just a small portion of the surface of the eye. Call that the lacrimal caruncle. What else do we have here? The sclera is the white part of the eye, of course. The colored part of the eye is the iris. I don't have that labeled on here, but iris. And then the margin between the eye, the sclera, and the iris is called the corneal limbus. Corneal limbus. It's limbus, what do I want to say to make it easier to understand? It's just the sort of milky ring around the outer margin of the iris. The dark spot in the middle is the pupil. That's how the light gets in. And then it's not labeled on here. I don't know. Do I have it here? Maybe. Sort of. It's a little better. Okay, so we've removed some of the anatomy here, the skin. And so we can see a few things. First, we see these lacrimal gland, the lacrimal gland that is in the superior lateral margin of the orbit. The orbit is the hole in your skull. This whole thing here is the hole in your skull that the eye resides in. So in the superior lateral margin of the orbit is the lacrimal gland, and it connects with the margin of the superior palpebra via the lacrimal gland ducts. The lacrimal gland ducts. The ducts of the lacrimal gland. This is where where tears are produced and then communicated with the margin of the eyelid, so that we can keep the ocular and on the underside of the eyelid, the palpebral conjunctiva. On the surface of the eye, it's called ocular conjunctiva, and on the undersurface of the eyelids, the palpebra, we call it palpebral conjunctiva, where we can keep that sensitive tissue moist. All right, the surface of the eye needs to be kept moist. There are a couple different types of tears. There are watery tears, and watery tears are the ones that are produced by the lacrimal gland. There are oily tears. Oily tears are produced not in nearly the same volume as watery tears, but they're produced by something not labeled here called the tarsal glands that live directly in the eyelid along the whole margin of the eyelid. It is the aperture for a tarsal gland that gets clogged, and then the tarsal gland infected. And when you have a sty, has anybody had a sty before? Yeah, chalazion. So yeah, and what next? So, but the watery tears are produced by the lacrimal gland, released onto the surface of the eye, and then when an eye blinks, I'm gonna do it here. It blinks from maybe I'm not doing it right. It blinks from lateral to medial. So your eyes don't go up and down like that. They kind of sweep close, brushing the tears across the surface of the eye from lateral to medial, and then I think I said this already, sort of slam dunks the tears into the lacrimal punctum. The lacrimal punctum. There's a superior and inferior lacrimal punctum. Anybody who has, again, maybe looked at your eye, or particularly a person who's put on, what is that stuff called, eyeliner or whatever, has seen those, the little dots on the superior and inferior eyelids towards the medial side. Right? That's where the tears that you generate moment to moment. So your eyes are always releasing a little bit of watery tears, and they are pushed into the lacrimal puncta, where they go down and join the ambient moisture in your nasal cavity. They pass through through these lacrimal canaliculus, plural canaliculi, just like the word that we ran across in the bone. It's the plural canaliculi. Just the little canals from the two puncta, and then they join the lacrimal sac, which is this little sac that sometimes, if you kind of push on the margin of your eyelid right there, you can get your eyes to squeak. That's you pushing, sort of pumping air in and out of the lacrimal sac. The lacrimal sac descends through the nasolacrimal duct and comes out beneath the inferior nasal concha in the nose. All right. So that's how tears work. What else do I talk about? All right, so we get a little bit of look at the external ocular muscles, the eye muscles, which help motion of the eye. And we'll see better pictures of them in a bit, but there are 6 of them. There's 6 of them. And 4 of them are pretty easy because they have the name rectus in it, and it's just the 4 cardinal directions: superior, inferior, medial, and lateral rectus. Rectus just means straight. They are straight muscles. So we can't see the medial and lateral ones here, but we can see the superior and inferior. There's 4 rectus muscles. And then there are 2 obliques. There is a superior oblique and an inferior oblique that come, as the name would suggest, obliquely inserting on the surface of the eye. We'll see better pictures of that in a bit. So, I'll make a point of something in there later. So here's one model. God, that's a crappy model. Talk about a lazy eye. I guess that's this person over here. We have much better models, though, now. So there's this model here in front of Haley that is probably the best model for depicting the external ocular muscles. One thing that you can see here, this would be the medial rectus. This is the superior rectus, cut and remove. This is lateral rectus, pulled aside. Inferior rectus here. Right down here, we have the inferior oblique. The inferior oblique is going to pull the eye. Let me make sure I'm getting this right. So, when my inferior oblique is contracting on my right eye, I'm looking up and laterally, superiorly and laterally, because it's pulling inferiorly and medially, and rotates the eye superiorly and laterally. The superior oblique is pulling the eye medially and no, it's pulling it inferiorly and laterally. They're both pulling towards the midline, but this one is pulling from the top surface, this is the bottom surface. So the superior oblique pulls this eye downward and laterally. The interesting thing about the superior oblique muscle is it goes around this hook here. It goes around a hook to redirect the direction of the muscle. That hook is called the trochlea. The trochlea. And that's a good thing to remember because that muscle has its own dedicated cranial nerve. So I told you yesterday there were 3 cranial nerves that control external eye muscles. And one of them is called the trochlear nerve, and it's just that muscle that it controls. And then the other is the abducens. The abducens muscle controls the lateral rectus muscle. It controls the inferior oblique and abducts the eye. And then the oculomotor nerve controls all the rectus muscles. I may be wrong. I may be switching those. I would have to check that. That's not something you're responsible for. But my point in telling you this is that each of those three nerves has specifically different, acutely different and distinct nuclei in the brainstem that controls them. Okay, so when you have a patient in the office that has some kind of cranial nerve palsy or perhaps a stroke, depending exactly on where it is, you're going to have very distinct patterns of oculomotor dysfunction. And understanding which of these muscles is innervated by which of those cranial nerves, again, can help you know at a glance where exactly the problem in the patient is. There's a tendency to rely heavily on imaging data, and of course you do imaging data or whatever, but the faster you know what you're dealing with, the better you are able to deal with it. So there are anatomical correlates to pathology that you're going to see, and knowing the anatomy is what will help you. All right. So there's a number of other eye models here. There's this one. There's another eyeball there. There's a couple other eyes. All right. So did I get everything here? I think I did. Yeah. Next. The cornea is the clear covering of the eye. And here's the corneal limbus, which is where the margin of the cornea meets the sclera, and it turns the coloration of the iris here a little bit milky. The pupil is the aperture through which light passes and comes in contact with the lens. And then the back surface of the eye is called the retina. This is where the photoreceptors are. There are 3 layers to the eye. If you think about the thickness of the eye, there are 3 layers to it. There is the sclera, and we'll go through this in lecture more. But there's the sclera, which is the fibrous tunic of the eye. It gives the eye its, it protects it. It gives it its physical shape. So there's the sclera. The next layer is called the uvea. I don't have the word on there, but you'll see it in a moment. UVEA. Uvea is the second layer, and that is the vascular layer. It's called the vascular layer of the eye. This is where we get the blood supply in. It's also where we have what's called the choroid. The choroid. So much like the choroid plexus in the brain, the eye has a choroid, which is going to produce the aqueous humor and the vitreous humor. So aqueous humor is in the anterior chamber, and the vitreous humor is in the posterior chamber. We'll go through those in a bit. And then the inner layer of the eye is called the retina. And the retina is considered to be the neural, N-E-U-R-A-L, neural layer of the eye. Neural meaning this is where the photoreceptors are. This is where the action happens. Well, this retina, as it runs towards the anterior portion of the eye, you have this margin where it kind of runs out. This is the limit of your field of view. All right? So if you take your hands and you put them out right at the margins, looking ahead, and you put them at the margins of what you can see, and you go in and out from what you can see and can't see in the periphery, the image of your hand is falling on the retina right around here somewhere. Okay? Whereas the thing at the center of your view is falling on this spot right here called the fovea. The fovea is the center of your field of view. It's the thing that you think you're looking at when you're looking at something. It's the center of your focus. All right. Is there anything else? This margin of where the retina kind of peters out and you see the underlying uvea or choroid, that margin is called the ora serrata, the serrated opening. Ora serrata, serrated opening. So here we can see the ocular conjunctiva on the surface of the eye, the palpebral conjunctiva. The word fornix, again, now we see that it's not just a structure in the brain, that it's actually a generalized term. And you can imagine why it might be called that, because it's where the ocular conjunctiva and the palpebra meet with one another. They join there. All right. So I'm going to point this out now. Here in the limbus, there's this little hole. And we'll talk about it in a moment, but it's called the sclerovenous sinus, or the canal of Schlemm. Canal of Schlemm. I love that. Don't know who Schlemm was, but he lives in immortality now. Okay, so I guess I already just went through all this, but I'm going to show it to you now because it does have a couple. It's a good diagram. So I said there were 3 layers. We have first this fibrous layer, the bulk of which is the sclera, the white portion of your eye, which then translates into the cornea, which is clear, the fibrous layer. Second layer is the vascular layer of the eye, the uvea, UVEA. And the portions of the uvea are the iris is part of it, what's called the ciliary body. Ciliary body, C-I-L-I-A-R-Y, ciliary body. And the ciliary body is principally responsible for controlling the shape of the lens, and we'll see how that works in a bit. And then lastly is the choroid. And the choroid is what produces the vitreous and aqueous humor of the eye. We'll talk about that more in lecture. And then is the neural portion. We have the retina. There's this neural layer, and there is a very thin single-celled layer called the pigmented layer of the epithelium. We'll talk about the function of that in lecture. Now, there are 2 cavities in your eye. There are 2 cavities in the eye. There's the anterior cavity and the posterior cavity. It's going to get a little more complicated than that. Let's see if I have a better picture. Yeah, okay. So here's the anterior cavity and the posterior cavity. The anterior cavity is broken up into 2 chambers, an anterior chamber and a posterior chamber. Earlier, like 5 minutes earlier, I misused those terms. I called this the anterior chamber and this the posterior chamber. This is not the posterior chamber. This is the posterior cavity. And the anterior cavity has an anterior chamber and a posterior chamber. The anterior chamber of the anterior cavity is in front of the iris, between the iris and the cornea. The posterior chamber is between the lens and the iris. Yeah, I wasn't being careful in my speech earlier. I realized that as I was saying it. So this is the posterior cavity. There are no chambers in the posterior cavity. All right. The posterior cavity is filled with the vitreous, vitreous, vitreous body. The vitreous body, has anybody dissected an eyeball like in high school or something like that? Did you get to pull the vitreous out of there? What did it look like? Yeah, it's this weird jelly ball, isn't it? And the aqueous humor, did you get into that stuff? Yeah, what was that like? Yeah, the vitreous body is solid. The aqueous humor is actually kind of a jelly, or between like it's like runny jelly. Not even quite that. It's very liquidy. It has a flow to it, whereas the vitreous is a solid thing that you can hold. The stuff up here, aqueous humor, is much thinner and can flow. What else do I want to point out here? Okay, so right here is the same dot, and this dot is really just a canal that runs in the limbus of the eye. So it runs beneath the limbus, surrounding the iris, the sclerovenous sinus, or sulcus circularis corneae, or the canal of Schlemm. It goes by a lot of different names. Sclerovenous sinus is the most common. This is how, just like the choroid is what produces the aqueous humor, and the choroid plexus produces CSF. We have in the brain the arachnoid granula, which resorb the CSF. Here, it gets resorbed into the sclerovenous sinus. All right, this is how we resorb all that and return it to venous circulation. All right. So, anterior cavity, posterior chamber, anterior chamber. What else do I have here? Okay. So the lens is held by these ligaments to the ciliary body, and we call this the suspensory ligaments of the lens. And the ciliary process itself, I'll have a better diagram of it soon. But there's a ciliary process and then a ciliary muscle. The ciliary muscle and the ciliary process together make up the ciliary body. The ciliary process connects to the suspensory ligaments of the lens, which then insert into the lens. The ciliary muscle is going to contract, and we'll see what that does in a moment. But the whole thing is called the ciliary body, the process and the muscle together. What else here? So again, you can see the fovea. Now, the students often get this confused. There's the fovea, which is at the center of your visual axis, but then there's also this optic disc. And the optic disc is where the optic nerve enters the back of the eyeball. This is your so-called blind spot. Your blind spot. You don't. Most people are not aware of this because your brains are actually smarter than you are. But it's about 23 degrees off lateral to your visual axis. There is a tiny little spot where you can't see. And what your brain does is it just like takes all the edges and smudges them in together. And we'll do something I should do it in the lab. But there's a little trick I'll talk about in lecture where you can test that. Yeah, I should move that to lab as well, shouldn't I? Okay, here's the ora serrata. Wait, was there anything else here? Nope. Ora serrata, nice picture of the ora serrata from the inside of the eye. Okay, so these models are the ones that you're going to want to look at. Here is sclera, cornea, choroid as part of the uvea. Here's the iris. Vitreous body. Here's the ora serrata in this really bad diagram. This is the lens, the suspensory ligaments of the lens attaching to the ciliary process. Here is the retina, the fovea, the optic disc, the ophthalmic artery and vein. All right. Yeah, so here's the better picture of the ciliary body, the ciliary process, the ciliary muscle, suspensory ligament, lens, iris, pupil, cornea, sclera, sclerovenous sinus. This is a better picture. Should probably just use that. Oh, yeah, the point being, the choroid, as is embedded in the ciliary body here, is releasing aqueous humor, the fluid humor, the aqueous, the liquidy humor of the anterior cavity. And this comes out, flows through the posterior chamber, out into the anterior chamber, and then is resorbed by the sclerovenous sinus. All right, you guys see that flow pattern there? Again, just same thing. It's probably cut out some redundant slides. Is there anything new in this? Nope, there's nothing new here. Yeah. Okay, on to the retina. So the retina has multiple cellular layers. And we have some nice slides of the retina. So I think here, yeah, you'll get to see some in a minute. But, so this is where the vitreous would be, right? Light is coming through the vitreous and going through the cellular layer, the neural layer of the retina. And the layers going from, I don't know what superficial or deep means here, but from interior to exterior are ganglion cells, bipolar cells, and that boundary of bipolar cells to ganglion cells has these amacrine cells which are mediating those connections. And then the next layer is bipolar to photoreceptors, the rods and the cones. And this layer is being mediated by these horizontal cells. Now these synapses right in here between bipolar cells, rods and cones, and horizontal cells are amongst the most sophisticated synapses in your body. Probably are the most sophisticated synapse in your body. Very, very complicated that we're only going to get a very small dose of in lecture today. It's a very high-end discussion talking about those. And then lastly, we have this pigmented epithelium, the job of which is to any light that does not get absorbed by the photoreceptors is absorbed by the pigmented epithelium so as to protect the delicate structures deep in the uvea, deep to the retina. So the job of all these melanocytes is to absorb any excess light. All right, that's just the anatomy. We're going to talk about the physiology in lectures, pretty much half lecture today. Okay, so here's a slide. Here we have, this would be, down here, this would be choroid. And here are the ganglion cells. Here are some bipolar cells. Here are the rods and cones. There are some, a few of these are the amacrine cell. No, these are horizontal cells. Amacrine cells are down here somewhere. But these are bipolar ganglion, bipolar rods and cones. And then this dark layer up here is pigmented epithelium and choroid. So what do you notice about this? So these are the nuclei of the rods and cones. These are the nuclei of the second layer, the bipolar cells, and these are some of the nuclei of the ganglion cells. What do you guys notice about that? I'm not asking for anything deep here. Just like, what do you see? Yeah, what do you?
Yeah.
Way more nuclei in the rods and cones. Fewer bipolars and much fewer ganglion, huh? So what does that suggest to you? Okay, well, certainly the whole system is very sophisticated. What it suggests to me is that there's some sort of convergence of information. That one ganglion cell is managing several bipolar cells, each of which itself is managing several rods and cones. There's significant convergence happening. All right. So the implication is that there is significant visual information processing that's happening right in the retina before anything even gets into the brain, before it even leaves the eye. There's a whole host of information processing that's happening right in the eye. All right. Vision, I guess the take home here is vision is profound. Vision is profound. And it's a very sophisticated process, being able to create an image, whatever that even means. That's more of a philosophical discussion. Okay, there's the eye. So the ear. The ear has three portions, the external, middle, and inner. And I'm just going to whip through the anatomy here, because we only have 20 minutes, and I'd like you guys to work with the models a bit. We have the external auditory meatus. There are two ear models as well. Where are they? They're out there. Oh, there's one there. There's one there. The external auditory meatus, your outside ear hole. External acoustic meatus or auditory meatus. And the deep margin of it, the medial margin of it, is the tympanic membrane, which is the beginning of the middle ear. And the tympanic membrane connects to the three auditory ossicles. And the auditory ossicle, so the incus, the malleus, and the stapes, or the malleus, the incus, and the stapes. Malleus hammer, incus anvil, and stapes stirrup. The stapes inserts into the oval window, and that is the medial boundary of the inner ear. All right. The middle ear, the other feature worth pointing out here, is it connects to the nasopharynx via the auditory tube or the eustachian tube. When you swallow, the cartilage of this is what you hear cracking open. So normally it's closed. When you swallow, the act of swallowing pops this cartilage open and allows pressure on either side of the tympanic membrane to stabilize. All right, so it normalizes pressure between the external outside air and the pressure inside the middle ear. This is why it becomes hard to hear if you have bad cold and the auditory tube, the pharyngeal tube, or the auditory tube, the eustachian tube, becomes blocked. Middle ear, the structures that we have here, this is the vestibule, and meaning here of vestibule is just the same as the meaning in a building or something, the entryway to the inner ear. It leads into all the different portions. The portion that gives us our sense of hearing is called the cochlea. Cochlea, just like conch, but there's no N, C-O-C-H-L-E-A, cochlea, this spiral snail-like structure. There is in the skull that's in the wooden box, which my diligent students have put away. It's over there, but the temporal bone, one of the bone, or maybe it's one of the skulls. Somewhere in the lab, there's a dissection of the middle ear or the inner ear. It's worth looking. But you can see the spiral organ there. This attaches to the cochlear nerve, and Vestibulocochlear nerve is cranial nerve 8. It has two branches, the cochlear branch, and then there is the vestibular branch. The vestibular branch, the vestibule of the inner ear, has these three semicircular canals that are all embedded in the petrous temporal bone. Okay, I think, last bit. So we have the oval window that the stapes fits into, and then there's this thing called the round window right next to it. And we'll see what those two things do in lecture. All of this can be identified on this model here. So we have the cochlea, vestibulocochlear nerve, cochlear branch, vestibular branch, semicircular canal. The vestibule is down in the model. You can see it. Here are the auditory ossicles. Take that whole thing apart and explore it. All right. So Malleus, Incus, and Stapes. The way you can remember this is you don't want to miss it. M-I-S, Malleus, Incus, Stapes, moving medial, lateral to medial, superficial to deep. Or Malleus meaning mallet or hammer. The hammer hits an anvil, or Incus is the word for anvil. And then Stapes is like a stirrup, it means stirrup. So you're taking the hammer, slamming on the anvil to make yourself a stirrup. There are some ligaments and named muscles. These are the two muscles. The tensor tympani muscle attaches to the malleus, and then the stapedius muscle attaches here to the end of the incus and stapes. Both of those muscles, their job is to dampen vibrations in this whole mechanism in the event of loud noise. So when there's loud noise, those muscles tense up and dampen whatever sound there is. You can see the round window here. If there's anything else. There is, I haven't really made a big deal of it because I didn't go into the facial nerve much, but the facial nerve does a lot. I did talk about it once when I was talking about taste. There's a branch of the facial nerve that comes through the middle ear. It's called the chorda tympani. The chorda tympani. It goes right over the ear drum. That's some anatomical minutia for you. Yeah, some inner ear ossicles. This thing's mine. I have those somewhere. I don't know if I brought those in this year or not. So vestibule, there are these different structures. Okay, so there is the utricle and the saccule, which each have these macula in them. The utricle and the saccule are two different sort of bulbs in the vestibule. There's this thing called the endolymphatic sac that dangles off of it. The inner ear is magical. It's like some kind of alien creature that's living in your head. I just find it remarkable that it looks the way it does. Endolymphatic sac is, so all of this is filled with a fluid. There's a bony labyrinth that you see here, and there's a membranous labyrinth that's running through it. And there's fluid, there's endolymph, and there's perilymph that are on either side of this membrane. The endolymphatic sac is how we normalize that pressure. So it's outside of the bony labyrinth and can expand or get smaller depending upon the pressure needs, the pressure differential between the endolymph parallel. Does that make sense? Okay. We're going to talk about how all this works in lecture today. It's one of the two main points. So then at the base of each of these three semicircular canals, there are these ampules, these three things called ampules. And the ampules, with in medicine, particularly back in the day when you were out in the battlefield or something, medicine would come in ampules. Something like this. And it would have your fluid down in here, and you would score this and snap it off. All right. Well, this these structures kind of look like the top to ampules to the anatomists, early anatomists that were naming this stuff. So that's what they're called. Inside the ampules are these things called the crista ampularis. The cristae ampularis are, there's 3 of them, one in each of the semicircular canals. We'll see some of those in a slide, and probably the next slide here. Nope, in a moment or so. We'll get to the cristae ampularis. So here's a cross-section, maybe of a semicircular canal or whatever. That whole weird alien structure that I was showing you, kind of looks like a nautilus or something, is called the bony labyrinth. The bony labyrinth. I had pointed out the membranous labyrinth, and I also mentioned the endolymph feeling the membranous labyrinth and the perilymph between the bony labyrinth and the membranous labyrinth. All right, does that make sense? You guys follow me? I know, huge amount of. This is one of the denser days for sure. So here is a not well projected picture of the crista ampularis. We definitely have some slides that show that. This is where we get our sense of directional acceleration. Like your body is tumbling in some plane or the other, and it's this crista ampularis that is returning you that information. So it's this little bump of tissue that has hair cells on it that are embedded in this gelatinous structure that's not showing up on this slide. They call it the crista ampularis. Inside of a semicircular canal, this whole thing is a cross-section of a semicircular canal in the ampulla. All right, so here's another cross-section, semicircular canal, cross-section semicircular canal. Here's the crista ampularis. There's hair cells that are embedded like this. You'll see better in a cartoon. You really can't see. Projections would be faint. I can see it like this, but there is this gelatinous structure that's coming up right here. It's called the cupula, C-U-P-U-L-A, cupula. The cupula. You'll need to know that so that we can talk about it in lecture today. Here's a good diagram. So, crista ampularis, hair cells. The whole thing is the ampulla of the semicircular canal. This gel matrix here is the cupula with the hair cells embedded in it. There's this support structure and sensory nerve that descends from it. What's going to happen is fluid is going to come across like this, bend this over, and depolarize these hair cells by bending the hair cells and opening the mechanoreceptors inside those hair cells. That's the shorthand story of how it all works. It's incredible to me. Feels like such a crime to go fast through it. The body is filled with so many wonders, isn't it? Here's a transect of the cochlea. This is the hearing organ. We will be talking about how this works in a little bit here, but if you were to look at this, imagine that it's a spiral chamber that's going all the way up, spiraling up to the top, and then connecting with another ramp that's in parallel to it and it spirals all the way back down again. Does that make sense? So imagine 2 spiral chambers that are spiraling up together and then joining at the top. It's another way to think about it. If you were down here, you could walk all the way around, come up here, and then here, and then here, and then here. And then up at the top, you would loop around into the other side of that spiral and come down to here, and then here, and then here, whatever. Okay? You'd eventually get all the way back down. In between is this third chamber. This is this third area. So this one originates at the oval window where the stapedius, the stapes, pardon me, the stapes inserts. This ends up at the round window. In between is this cochlear duct. This whole structure here, this cochlear duct, this is going to be the hearing apparatus, and we're going to see how that works in a bit. It names all these membranes. We're going to have to learn them, but we have a better diagram in a bit. So there are slides that show all of this. Let's come back to that. I want to look at this in more detail. I called this the cochlear duct. They call this the scala vestibuli or vestibular duct. This is the one that goes to the oval window. And the scala tympani or tympanic duct. This is the one that goes to the round window. All right? The two are separated by the cochlear duct. So tympanic duct, vestibular duct, cochlear duct. And now we're going to talk about this thing. It's the organ of hearing that we have. But while we're still out, we see that each of these turns is served by what's called a spiral ganglion. Called spiral because it's part of the spiral. It's a ganglion that we have a cross-section of it here, but really this ganglion spirals around. It's a long spiral that goes through the whole cochlea, right? So this continues down in the bone, loops around, and it comes back to here. It's one long spiral ganglion. That make sense? Yeah, so let's look at a closer picture of this. I have one here. There we go. So the membrane between the scala tympani, or the tympanic duct, and the cochlear duct is this basilar membrane. And then above it, above the basilar membrane, are these hair cells. There's two groupings. There's outer hair cells and there's inner hair cells. Those hair cells are in close contact with this little lip that sticks over it called the tectorial membrane. We'll talk about how this works exactly in lecture in a bit, but this is the anatomy for it, the tectorial membrane. I have a beautiful model of this in front of Liam and Maria over there that you can see. You should spend some time with that model. So the membrane between the cochlear duct or scala media, the cochlear duct, and the scala vestibuli or vestibular duct is called the vestibular membrane. All right, it's this little vestibular membrane. Here's a kind of weird tectorial membrane. It's been messed up in the section. Here's the basilar membrane, hair cells, inner and outer hair cells, spiral ganglion. Let's go back to the zoom out. So vestibular membrane, basilar membrane, vestibular duct, tympanic duct, cochlear duct, spiral ganglions are here. Oh, that's it. Okay. A lot of anatomy, but we saved ourselves some time for lecture by going through it all in detail here. Spend a little bit of time just like strategizing some of the models, take a look at some of the models, and then we'll head down to lecture to talk about this. Are there any questions?